So I'll get straight to the point of where we want to go today, namely our introduction of a practice known as Predictive Toxicological Approach to nanomaterial safety testing as an alternative test strategy. And this, of course, is derived from the perception by several countries, including the US, where the National Academy of Sciences became concerned about the number of untested chemicals. Of more than 70,000 industrial chemicals, fewer than 1,000 have ever undergone safety evaluation comprehensively, and the backlog is creating problems. Now, nano, since its advent, is also moving rapidly, and so the idea then is of a 21st century vision for chemicals, where we move away from one material at a time to wide coverage of several toxicants, instead of a descriptive approach that is done in animals primarily, a robust scientific platform for screening, doing it predictively, and I will describe what we mean by prediction, being able to do it high throughput, but importantly to have a connectivity to in vivo or animal research. I personally, my own viewpoint is we can speed things up, we can reduce animal use, we can gather information, but we're not at a point yet to replace animals. Now, of course, interestingly enough, the man who cloned the Genome Project and leads our NIH, when his laboratory moved from after the gene was cloned, Francis Collins invested a lot of their own personal infrastructure in terms of research into a toxicogenomics initiative, where the vision was to move away from, human studies 1 to 3 per year or standard testing that is being done in rodents, to a science where alternative test platforms are being used, including computational toxicology and critical toxicological pathways. So with that vision, of course, then what is happening and have been implemented already by EPA in their ToxCast program, and then in association with the NIEHS, FDA, and the National Toxicology Program, the Tox21 initiative, this is real-time data gathering that is ongoing. And it comes from the era of the genome, proteome, epigenome, transcriptome advances, where you can move from molecules, genomes, to cells, to whole organs, disease, pathology, and where there's a huge availability of in vitro and in vivo data sets that are being integrated. And in my perspective, at the moment, an exciting advance in science, but we need to recognize that at the moment it is so data rich, and that we are still at a point where we are collecting what I would say signatures. And as an example of the signatures, if you see the width of this program, these are just the disorder categories being probed by the EPA ToxCast program, where in different areas huge amounts of data are being generated, but not yet ready for implementation in terms of making toxicological decision making. And just to show you how the breadth of this information is being used, is that just in phase 1 ToxCast, using 600 different in vitro assays across 60 in vivo endpoints and 84 different statistical classification methods for the high throughput screening on the first phase, about 300 different ToxCast chemicals, is that you can see the breadth of discovery, but you could also see the complexity in order to integrate the in vitro and in vivo data. And at the moment, that is proceeding by many different classification algorithms. So considerable complexity, but I hope to indicate that out of this exercise, I think will emerge a very rich infrastructure that indeed will move that 21st century incrementally. So here is our idea then of a predictive toxicological paradigm that sort of mirrors what I've just shown you, but on a much simpler basis. In that we advocated, when we began the CEIN, a proportional weighted discovery, where we said it's easier to collect data at cell biomolecular endpoints, where you can collect up to 100,000 data points, and I'll show you one or two examples of that today, as compared to animal testing, where if you're fortunate, and say for instance in rodents, you're lucky if you can move up to 100 animals in a single experiment. So we advocated making the discovery at biomolecular and cellular endpoints, but through choosing what to test based on mechanisms of toxicological injury that play a role in the pathophysiology of disease, that you can do your discovery here and build structure-activity relationships and dosimetry that also pertain to the in vivo outcome. Once you have confirmed the in vivo outcome from the prediction making from in vitro, you can then proceed to do a lot of discovery at this endpoint, occasionally going forward to, again, checking that the in vivo system gives you a reality check. And so for this reason, we maintain animal use, but we've reduced them, and I hope to show you one or two examples. Now, in order to launch this discovery for nanomaterials, introducing hundreds and thousands of new products, constructs, and a wide range of properties, we could not wait for all these materials to emerge. So we engaged in a science of making nanomaterial libraries of different compositions and accentuated physical chemical properties to kickstart the discovery process. And so to put that just into proportional perspective, compared to the phase 1 toxicology, which is a very high data-rich discovery. So if you talk about high throughput here means hundreds to thousands of chemicals in one sitting. The predictive tox that we're implementing, we're talking here about single-digit to double-digit number of materials, but still much more comprehensive than what has been done by classical testing, where we then use high throughput discovery of pathways of toxicity to generate tox data, and from there build the science of structure-activity relationships to make our predictions. Now, the tools that are required in order to participate in this is choice of materials. So originally we were guided by 13 different OECD selected materials. We had to implement discovery of nanomaterial properties, rigorous physical chemical characterization. I'm not going to go into all the details, but interestingly, this sort of disclosed that, you know, there are intrinsic material properties that, as acquired, the size distribution of the materials, etc. Extrinsic properties, the alteration of those properties, if you put them in a biological media, that is, for instance, the zeta potential and the protein corona and the charge on the materials. And then after you've collected those data, that also hope to show you that there's a third group of properties that have emerged, those that come out of the structure-activity relationships that often do not contain any of those 14 or 15 major properties that we set out to characterize. To build the platform, we implemented high-throughput screening, where you will see some of our infrastructure today to do it at cellular level. We've also got one for zebrafish that we do for organisms. The acquisition through the activities of what we call our Theme 1 under Jeff Zink's direction of initially nanomaterial compositions of the different types of materials that OECD selected. And once we've gathered data, is then accentuation of properties such as size, shape, aspect, ratio, crystal structure, band gap, dissolution chemistry, etc. in these materials to build libraries of materials that can be used for high throughput screening with pathways of toxicity, readouts, variety of different readouts, computational analysis, and heat map ranking in order to provide an initial decision-making tree of in vitro hazard ranking that you can use for further implementation. So I'm just showing you here an example of libraries. This is a silica library that was tested and different to what was predicted. The crystalline silica, or quartz, is the most hazardous form of silica. Out of the screening analysis actually emerged that it is the properties of the silica ring and the surface display of silanol groups that is the most important. And so Jeff Zink and Jeff Brinker built a very sophisticated library to investigate that possibility. For carbon nanotubes, more than 50,000 different combinations possible if you take all the physical chemical characteristics. I show you here from a single multi-wall carbon nanotube a series of surface functionalizations that was introduced to cover that one group of what does surface functionalization do for carbon nanotubes. For a material like cerium oxide, where preliminary research indicated that in addition to redox properties, the length scale of the cerium oxide may make a difference. The introduction of a library that varies from spheres to rod-shaped materials of different aspect ratio. You will be introduced today to our high-throughput facility, a superb facility established here in the CNSI, where we have a variety of different readout methods. A major advance from our research is the pathways of toxicity, of which we've now, through discovery, covered about 8 to 10 different pathways of toxicity that can be used for high throughput screening. Oxidative stress, metal dissolution, cationic nanomaterials, and the interaction with different phases of the cellular machinery. A pathway that is triggered by lysosomes for long aspect ratio material, photoactivation, and then there's a property of silica libraries where the silanol display can lead to membrane injury as well as triggering the lysosomal pathway. And then finally, what Urim will be talking to us about the implementation of high content data exploration and structure activity relationship modeling, in which he will explain to us the various different stages in which the information is collected and move through from pre-processing to data exploration to the establishment of structure activity relationships, quantitative structure activity relationships, and making prediction on that basis. So just as one example of such an exercise of nanomaterials that can cause hazard, one of the major hazards is the ability to generate oxygen radicals. And I don't have time to go into all the different categories, but for metal and metal oxides, their semiconductor properties play a very important role. And then in this flow chart of how we've been able to analyze a relative large number, 25 different metal oxides, by use of this exercise, is to make use of their redox properties, the band gap of these materials, a cellular screening assay that allows us in a multi-parameter fashion to measure oxidative stress responses that range all the way from early perturbation of defense mechanisms to inflammation to ultimate cytotoxicity, to implement this with robotics in a high throughput screening process, data exploration, of which I'll show you one format, a heat map where you can see out of the 25 materials, 7 of the materials have the red spot, which means that they triggered one of these toxicological responses, using the hazard ranking then in an animal exploration in order to quickly move through all 25 materials in the matter of a few weeks instead of the approximate 2 to 3 years it would have taken if work was done by conventional means. So out of the screening of the materials that I've shown you, materials who have a conduction band that overlap with a similar energy level in the cell, known as the redox potential, we could then demonstrate that the in vitro screening, moving this to animal studies where we deliberately made use of materials in the overlap, materials outside, to be able to rank all these materials by acute inflammatory effects in the lungs of these animals. And this provided therefore a hazard ranking that can then move up to the next phase of animal testing, namely inhalation studies. So out of the prediction toxicology that followed from this exercise, from libraries to high throughput screening, 25 different metal oxides, everything that's the red is a potentially toxic material. We could do a regression tree analysis where zinc oxide, copper oxide in that group was due to high dissolution rate, and from the rest of the materials there were 5 materials with an energy band gap overlap with cellular redox potential that allowed the generation of oxygen radicals and oxidative stress that played itself out in the animal model by the initiation of pulmonary inflammation. The rest of the materials which do not form, this is the band gap energy, that do not fall in this range, did not show their toxic potential. So similarly for carbon nanotubes, you just take the large range of properties of carbon nanotubes that function in a hierarchical fashion in order to introduce injury. Theoretically, you can calculate there's more than 50,000 different formulations that can follow if you systematically vary these properties. The initiation then of a predictive tox screening, in this case that was determined by pathophysiology of disease, that the idea is that of many potential adverse health effects of carbon nanotubes, its ability to introduce hazardous properties in the lung, which leads to lung fibrosis, involves a concept out of pathophysiology of lung fibrosis where there's cooperation between a nest of cells, macrophages, epithelial cells, which when introduced to macrophages with potential, to multiple carbon nanotubes with potential hazardous properties, can trigger a march of events that you can measure in cells as a series of cytokines and growth factors that's being produced, and that also faithfully reproduces, if you introduce this material in the lung, the production of the same growth factors, with the exception that as in a tissue culture dish, the different biomarkers do not all appear on the same day. It's a chronic march of a disease event where, for instance, the interleukin-1 beta from the macrophage appears very early on and then gives rise through progressive interactions with the epithelium to other growth factors. And then to bring that to a structure-activity analysis for the production of the interleukin-1 in the macrophage, we could go down to a biomolecular level where the material characteristics, such as the long aspect ratio surface catalytic chemistry, plays itself out in a deliberate arrangement with the lysosome, where it detects first material properties that will internalize the material, and then catalytic surface properties that will damage the lysosome, which you can image. You see a dye here, you see intact lysosomes. After the introduction of hazardous carbon nanotubes, you can see the dye is released in the cell, and it then switches on a little piece of machinery known as an inflammasome to start producing the interleukin-1. So if you integrate all these discoveries, so I'm now showing you the series of carbon nanotubes with surface functionalized properties, we could have a display of various different surface functionalizations. We could do cellular experiments to show, if you look at the height of the bars, that indicates their cellular injury potential. If you look at the bottom end, this is the lung hazard potential, so you can see how the size of the different bars fit together in order to show the prediction outcome. And then if you want to go down to a molecular structure-activity relationship, you will also see that material number 1, here, high bar, injured the lysosome. Material number 2, which is a carboxy function like carbon nanotube, had very little hazard potential. And we could therefore arrange a grouping of carbon nanotube to say if you surface functionalize them, those that have cationic surface charge have to have special considerations compared to those that have a anionic surface charge. So through our predictive toxicological exercise, we have then moved through now more than 200 different materials, where through this exercise of high throughput or high content screening, we could make predictions, we could hazard rank, we could take them to the lungs of the animals, we could show that the hazard ranking is predictive of the outcome, and we could take the hazard ranking then in the exercise as we apply it to make decisions on which materials at facilities that we don't have, namely inhalation facilities, could be tested further. And in her talk, Eileen will also highlight here the important work that they do of NIOS. So in translation of our work from the in vitro predictive assays that rank the property of materials versus well-defined positive and negative controls that we built in our own libraries, we can now take any nanomaterial, carbon nanotube for instance, tell you its relative toxicity potential compared to positive and negative controls. We can develop quantitative structure activity analysis, in which we can do in silico predictions. We can do a tier 2 testing, which is short-term in vivo testing. This is usually done by non-physiological exposure, such as oropharyngeal instillation, but nonetheless very valuable to take the groups of materials collected here, further characterize them so that we have in vivo hazard ranking potential. Because we can make these predictions, it focuses and limits the number of animal studies. We can validate the mechanisms, the potency in a group, and then from the in vivo hazard ranking potential that is built around this concept of pathways of toxicity and pathophysiology of disease, we can then move on to tier 3, where we now, for more expensive studies, be able to select within a group the materials that we tested from the tier 2 category, and we can do dose response extrapolations, and from there move on to getting occupational exposure limits and read across that can be potentially implemented for regulatory decision making. So Eileen will explain the importance here of then a critical development of science where we can use dose response extrapolations, even although we don't have human exposures, but use workplace exposures in humans to calculate dosimetry from the tissue culture dish to the animal lung to the human lung based on a metric such as amount of materials per unit surface area in these exposures. And then being able to use exposure environment data deposition in the lungs of humans to back calculate what a potential relationship of a dose in a human to an animal to an in vitro exposure could be. Where I think our work currently fits in to the ToxCast and the other programs is they are collecting signatures of toxicity that is currently being mined for pathways of toxicity as one outcome out of those signatures. We have implemented this on a much more simplistic scale, but still effective from the choices that we've made to allow high throughput screening, hazard ranking, in which we could prioritize animal experiments, and through further work in the tiered approach that I've shown you is now move this into risk translation exercise. And then finally can also use this for safer design of nanomaterials because we now understand properties. So in summary, the advantages of the predictive toxicological approach and rapid throughput screening is that rapid screening of large material batches, reduced and streamlined animal use and cost savings, a systems biology approach and a robust strategy to allow quantitative hazard assessment, in vitro and in vivo hazard ranking that can be used for focused inhalation dose extrapolation, and a tiered risk translation approach. Implementation of computational modeling and SAR analysis. Jerome will tell us more about this, and assist the development of a 21st century tox paradigm that may also be helpful to chemicals. And then there's also important, in terms of consideration of this workshop, the pitfalls of this, namely the fact that this will require acceptance by regulators and industry under the current statutes and the rules of engagement, and we hope to talk about that in the afternoon sessions. The fact that you use robotic technology, which often is expensive and not everybody can afford it, but again, out of the spin-off could be miniaturized lab-on-chip approaches that incorporate these readouts. In vitro to in vivo disconnect, that's often mentioned, that if you do just discovery in cells, that this is not whole animals, we agree with that. That's why we do the animal use or maintain that. The fact that you could generate lots of false positive and false negative, we think that we can offset that by maintaining animal testing and using our mechanisms of injury, and also building up our library of materials in which we have positive and negative controls. Non-specific and off-target effects, that means that there may be things that contribute to toxicology that we didn't think about and not include in our screen. That still is a possibility, but here again, the systems-wide discovery that is currently ongoing by ToxCast will be helpful to fill those gaps. No real-life exposure analysis, that's true, but is offset by what Eileen will show you by using benchmark materials and doing dose extrapolation. Test predominantly acute toxicological events, that's what we frequently hear. That's true to some extent, but I've shown you for carbon nanotubes that we can test an acute event which contributes to a chronic pathophysiology of disease outcome, so that even though I do it in a test tube in one day, I can predict what's going to happen in the animals over weeks to months. And then finally, the rigorous validation requirements, which to implement alternative test strategies is an exercise that traditionally, through the various different agencies and authorities that use them, that will take time to move it from development through regulatory acceptance. And then finally, I want to just throw up this slide in order to acknowledge a multidisciplinary CEIN, consists of more than 100 different people in the center, lots of contributions, and I show here a few of the people who generated the data on the lung tox screening. And we'll move on to any questions if you have.